Conjugation filter mating technique is a process in which bacterial conjugation mediates contact-dependent transfer of DNA from donor to the recipient bacteria. To spread the virulence and resistance plasmids, it is a 5 days process. On day 1, we do clean the surface area with the 70% ethanol. We clean all our equipments with the 70% ethanol as well. Then it's time to label your plate for the strains you are going to streak on them. On day 1, we only fresh our cultures in which conjugation is going to be meant. So here I am steriling the loop in the red hot area on a burner, and then I'll wait for loop to be cool. I'll pick up the inoculum and streak it on the new plate, as my bacteria is gram negative, so the plate is of McConaughey. Here I am picking the bacteria inoculum and I am going to streak it on the plate. I have 2 strains, so I draw a line in between the plate and I will streak both on the same plate, and then it's time to put them into the incubator for next 24 hours at 37 degrees Celsius. On day 2, it's time to inoculate the antibiotics into the LB broth. In the epitrophs, I have the stocks of antibiotics. So I am actually using 2 strains. So I have 2 antibiotics. One is of tetracycline and the other stock is of ceftriaxone. And I am going to inoculate these antibiotics into the LB broth. 100 mg stock has been picked up from the epitroph and I added into the falcon tube. Same procedure will be performed with the second antibiotic. I am checking the epitroph so that the antibiotic is thoroughly mixed in the epitroph, and now I will inoculate the LB broth with the second antibiotic. Now I will inoculate these falcon tubes with the respective strains. The one strain is D and the other is R. So in one falcon tube I will inoculate D strain, and in the other falcon tube I will inoculate the R strain. So in this way I will get a broth, I will get a culture which is of the strain which are antibiotic resistant. Like one is tetracycline resistant and the other one is the ceftriaxone resistant strain, and I will get their cultures in the broth form. After inoculum, these falcon tubes will be placed into the incubator for next 24 hours at 37 degrees Celsius. Here I am adjusting my falcon tubes in the incubator and I will wait for next 24 hours. On the day 3, it's time to centrifuge your falcon tubes at 14,000 rpm for 10 minutes. Make sure that your centrifuge is balanced. The culture will be pallet down and a supernatant will be discarded then. And I'll discard the supernatant from both falcon tubes. Now it's time to wash this pallet with the PBS solution. I will add 10 ml PBS solution in each falcon tube and each pallet will be washed twice. Now this pallet is of the strain which are resistant to their respective antibiotic. The D strain is resistant to tetracycline and the R is resistant to ceftriaxone. Here I am washing this pallet with the PBS. Now it's time for the other pallet to be washed with the PBS solution. After washing, I will suspend the pellets in the 10 ml PBS solution. You can suspend the pellets in LB broth as well. Now I will add 400 microliter of this solution into one epitroph from one falcon, and will add the 400 microliter solution of the other falcon into the another epitroph, and then I will make a third epitroph in which the culture is from both falcons, like 400 microliter of tetracycline solution and 400 microliter of ceftriaxone culture will be added into the single epitroph, and the total solution will be of 800 microliter. It's time to get the plates ready for filter mating. I have 3 plates. One plate is labeled with the strain of tetracycline, the other would be of ceftriaxone, and third plate is for conjugate I will plate filter papers on all 3 plates one by one. Remember that these are simple LB broth plates. No antibiotic is being added into the LB broth while preparing these plates. So I will now add the filter paper. I will take the filter paper with the forceps and will gently place it on the plate surface. You cannot invert the plates while performing this activity. Now I will pick up the 100 microliter of the culture of tetracycline resistant bacteria and will place it onto the filter paper of the respective tetracycline plate. As you can see that I am slowly pouring the solution onto the filter paper. Now I will take the ceftozyme culture and will place it into the respective plate. It's time for the third plate now. So the culture, which is the mixture of both the cultures, will be poured onto the last plate, which is the transconjugate plate. It's time to prepare the plates for day 4. I will do it on the day 3 to save some time. So I will prepare 3 flasks of the LB broth, and in each flask I will add the respective antibiotic. Like in the first flask I am adding the antibiotic solution of tetracycline, again 100 mg, and in the flask 2 I will add the ceftozyme solution antibiotic, and in the flask 3 I will add both antibiotics. Here I am pouring the LB broth into the plates on the day number 4. I will pick up the filter papers on which the culture has been grown and I will add them into the new falcon tubes. Each filter paper will be added into the separate falcon tube. Labeling is very important. You have to label the falcon tubes plates correctly to avoid any confusion, and you have to be attentive while performing all this activity. Filter paper from all the 3 plates will be removed and added into the falcon tube. Meanwhile, I will prepare some dilutions. Here I am adding 900 microliter of PBS solution into the 3 set of F1 drops. One set will be reserved for tetracycline, one set will be reserved for ceftozyme, and the third set will be reserved for transconjugate. I am just adding the PBS solution into the falcon tubes. I will add the culture later on. Before adding the culture, there is a step which I will be showing you later on. Now this is that step. These are the same falcon tubes in which I have added the filter paper. Now I am adding the PBS solution onto the filter paper to wash the culture off from the filter paper, so that the culture which is grown onto the filter paper is suspended into the PBS solution. Now the same step of washing will be repeated with the other 2 falcon tubes which contain the filter papers. This is a falcon tube of the transconjugate tetracycline and ceftozyme. I am washing it with the PBS solution. And now this solution will be added into the dilutions which I have prepared earlier. 100 microliter of this solution will be added into those 900 microliter PBS solution dilutions. Here you go with that step. I'm adding the tetracycline culture into the respective dilutions, and then the ceftozyme cultures into the respective set of dilutions, and the culture of ceftozyme and tetracycline into the respective set of dilutions. 3 dilutions will be picked up from this step and will be spreaded onto the LB broth plates which I have prepared earlier. So I have taken the culture dilution and I am going to spread it onto the LB broth plates. The plates contain the respective antibiotic in it. After spreading all the dilutions onto the plates will be placed into the incubator for 24 hours at 37 degrees Celsius, and on day number 5 you will note down the results. The plates will be observed for the bacterial growth. If there is a growth into the plate of ceftaxime and tetracycline, it means that conjugation has taken place and you got positive result for conjugation. Now as you can see that these plates are labeled as C plus T, which means that they have ceftaxime and tetracycline both in them, and the growth on these plates indicate that we have successfully got a conjugate which has the both ceftaxime and tetracycline resistance in it. And that was our sole purpose to design this experiment, so that we got a single bacterial strain which has resistance to both genes. हमारे पास individual resistance already present थी. We got a strain which has ceftaxime resistant. We also have a strain which has the resistant for tetracycline. But we want that कि हमारे पास कोई एक ऐसा strain आ जाए which has the resistance to both genes, and that is being done by performing conjugation. Specifically, filter mating conjugation technique has been used in this experiment to get a single X conjugate. 5 dilutions have been prepared to culture the bacterial strain on the plates, and as you can see that even on the plate of the dilution 10 to the minus 8 there is sufficient growth on the plate, so it means the successful happening of conjugation in this experiment. Thank you so much for watching this video, and if you have any question you can ask in the comment section.